Today I'm going to present vectors for eukaryotes. So we have already studied in our course In-Vitro Biotechnology the vectors for the prokaryotes. So today we are going to embark on chapter number 7, the vectors for eukaryotes. As this is a big chapter, so I have divided it into 3 parts. So the first part we're going to learn why do we need to clone genes in eukaryotic hosts? This would be our first learning objective. Then, what are eukaryotic vectors? And then, how useful is yeast in cloning eukaryotic genes? And which types of vectors can be used with yeast as a host? So, let's just move on. Okay, so our first question first. Why do we need to clone genes in eukaryotic hosts? So to understand this, we will have to recall a few things. Like we will have to first see that why do we need a eukaryotic host? Okay, so we will have to revert back to our aims of cloning, and we will just describe briefly here. If we are using a prokaryotic host, what is our aim of cloning? Our aim of cloning here is to study the basic genetic structure and function. Okay, while if we want to use a eukaryotic host, so what we are looking for when we are developing a cloning experiment, we actually want to study a gene, and more precisely we want to see the expression of a gene in terms of protein production. Most of the time it's a pharmaceutical protein like a hormone such as insulin. Okay, or sometimes our intention or our aim of cloning is to make a genetically modified organism such as a plant with a herbicide resistant gene. Okay, so which kind of host we want to select, it basically depends upon our aim of cloning and what outcome do we need from our cloning experiment. So coming back to our question that why do we need a eukaryotic host? So the answer lies in two problems that we come across when we are using a prokaryotic host. What are these problems? So the first problem is the difference in the genetic structure of a prokaryote and a eukaryote. So you can see here, so this is a prokaryotic gene structure. You can see that there are multiple genes present together on a prokaryote, and such genes we already know that they are called operons. Okay. Now, when such genes are expressed, so you see there is a non-coding region, a regulatory region in the start, and then we get a polycistronic messenger RNA. What do we mean by a polycistronic messenger RNA? A polycistronic messenger RNA codes for more than one gene, and eventually it leads to production of multiple proteins. While on the other hand, if you look at a eukaryotic gene is basically split up into coding and non-coding regions. We all know that coding regions are called exons, the non-coding regions are called introns, and the difference doesn't end here. The next point of difference is transcription. When the transcription happens, what happens that after transcription, we get a monocistronic messenger RNA, the one which codes for a single gene, a single protein, and then later on, this monocistronic messenger RNA is modified, and such modifications are termed as post-transcriptional modifications. The post-transcriptional modifications include the splicing. Splicing means the removal of these introns and joining the exons together, and then the poly-A tailing. A poly-A tail is added at the 3' end to protect the messenger RNA from being degraded and by the exonucleases, and then a 5' G cap is also added. Now, if we see such kind of modifications are not seen in a prokaryotic gene. Okay, so obviously if we are going to clone a eukaryotic gene into a prokaryote, so a prokaryote would not be able to do such kind of post-transcriptional modifications, and eventually it will not uh, give us the desired outcome. Okay, this is the first thing. The second thing is Post-translational modifications. Okay, now post-translational modifications do occur in the prokaryotes and in the eukaryotes, but the nature of post-translational modifications in prokaryotes and eukaryotes, it varies significantly. Okay, now if we see here, there are different kinds of modification. What kind of modification? Amino acid modifications like deamidation, elimination. Then cleavage means proteolysis, removing some parts of the protein to make it active. Then ubiquitination, then UBL protein conjugation, and then we can see like addition of complex molecules like AMP, ADP ribosylation, adding of glucose, so glycosylation, isoprenylation, and then addition of certain chemical groups like hydroxyl group, phosphate group, acetyl group, and methyl. Now all of these things, they are required to make a eukaryotic protein fully functional. Okay. Now similar modifications, they do not occur or exist in most of the prokaryotes. Yes, prokaryotes do have post-translational modification, but they are less common and mostly of a different nature. Okay, for example, bacteria have different system for N-linked and O-linked glycosylation, which is a very vital post-translational modification in most of the eukaryotic proteins. And also the bacteria do not have widespread serine and tyrosine phosphorylation, and you see serine and tyrosine phosphorylation is of vital importance in the eukaryotic proteins. Okay, so we have two problems here. We discussed the two main problems for which sometimes when we want to express our gene of interest, or when we want our gene of interest to end up giving us a target protein, then we will try to eventually clone our gene of interest in primarily or secondarily in a eukaryotic host. Okay, why we prefer a eukaryotic host? Because eukaryotic host can provide a native environment to the gene so that it can be expressed, post transcriptionally modified, and post translational modification of the proteins can be carried out. Okay, now let's move on. So now we have selected a eukaryotic host. So what kind of vectors will we use so that we can clone a gene in a eukaryotic host? Okay. Now let's go back to the classification of vectors, and you see the vectors can be classified on the basis of the cloning experiment aim. Like, do we want to clone a gene or we want to express a gene? What do we mean by just cloning a gene? Cloning a gene means that making more copy numbers so that we can study a gene. Either we study the structure of the gene or we study the function of the gene. Okay, when we want to study the structure of the gene, mostly we are using the cloning vectors. When we want to study the function of the gene, we want to express the gene, so we are using the expression vectors. And if we are having both the aims, then we are using the shuttle vectors or the vectors which can fulfill our needs. The second way to classify the vectors is on the basis of the host. Okay. Now, so on the basis of host, we have vectors for bacteria, like we studied in chapter number 6, the vectors for E. coli, and vectors for yeast, which we are going to study in chapter number 7 now, vector for plants and vector for animals. There is a third method of classification of vector as well, and that is on the basis of the host. So vectors can be termed as the prokaryotic vectors and the eukaryotic vectors. Obviously, the prokaryotic vectors and eukaryotic vectors will be different from one another in certain things, like origin of replication, okay, like signals for the protein production, like start signal for gene expressions. So all these things will be different. Okay. Now let's move on to our eukaryotic vectors. Okay, what are eukaryotic vectors and why do we use them? Okay, so eukaryotic vectors, they are used basically for cloning and expressing the genes in eukaryotes. So eukaryotes, which type of eukaryotes we are going to study? Yeast, animal, and plants. Okay, so basically these vectors are constructed using the things which are available in these hosts. And what is that? First thing first, plasmid. Okay, so plasmid, you see, mostly we have seen that plasmids are present in bacteria, but luckily there is a 2 micrometer plasmid present in the yeast. Okay, then in case of plants, plants are associated with some bacteria like Agrobacterium tumefaciens, which contains the Ti plasmid, and Agrobacterium rhizogens, which contains the Ri plasmid. And all of these plasmids have been explored to develop vectors. Then, secondly, like bacteria is attacked by several viruses, plants and mammals and insects, they also have several viral pathogens. Now, these viral pathogens can be used to develop vectors for plants, mammals, and insects, respectively. Then, another unique type of vectors are based on the transposons or the jumping genes. Okay, so the transposon or the jumping gene which we are going to study in this chapter are the P elements and P elements from Drosophila, and these have been used to develop a unique type of vector. Okay, now, so let's come to our third learning objective. Why yeast? Okay, the chapter 7 begins with yeast as a eukaryotic host for cloning eukaryotic genes. And also when we are looking at yeast as a host, so we will look for the vectors which we can use for yeast. Okay, now, so why yeast? Why do we pick up yeast? We all know that yeast are simple eukaryotic microorganisms, and mostly they are unicellular, and however there are some species which are multicellular. Size is fairly small, 3 to 4 micrometer in diameter. They reproduce asexually by mitosis and by budding, and they are termed as chemoorganotroph. Mostly they do not require a very complex nutrition to grow them, and we can make simple media in the lab to grow the yeast. Okay, now often the yeast is isolated from sugar-rich materials like the skin of fruits and berries, but they are also isolated from soil and insects. Okay, so here is a scanning electron micrograph of a yeast. You can see a budding yeast here. Okay, now, so this article I have taken from Nature. It was published in 1978, and this was the first report about the eukaryotic gene cloning and expression in yeast. And here in this paper they have explained that why do they use yeast. Okay, and if you just zoom it in and then just read over here, so you will find here that basically most of the eukaryotic genes cloned at that time, with the exception of a few, do not seem to be expressed in E. coli. So you see why they moved on from the E. coli, because the eukaryotic genes which they were trying to clone and express at that time were not properly expressed in E. coli. Okay, and this gives the rationale to use yeast. Okay, so there are several advantages of yeast. So what are the advantages of yeast? Yes, obviously it's very simple. Okay, can be unicellular and multicellular as we saw. Then another very big advantage why we choose the yeast is that its biology, its genetics, the gene structure and physiological needs, requirements all are known. Okay, it's very easy to grow in cultures as well as in the bioreactors if we want to make protein at an large scale or at an industrial scale. Now, also it has a natural plasmid, so the scientists were interested in the 2 micrometer plasmid present in yeast so that they can develop vectors. And the third thing, which is the fifth thing, which is really very important, is that they are generally recognized as safe. The yeast, like Saccharomyces cerevisiae, we are using in our homes every day, like in making of bread. Okay, they are also used in fermentation and stuff. Okay, so let's just have a look at the yeast 2 micrometer plasmid. Okay, now this is the yeast 2 micrometer plasmid, and you can see here it has a region of replication. The size is pretty small, 6 kb, and it has a flip gene FLP, which transforms it from one form to another form. Obviously, predominantly there is a form A, and then there is a form B, which is not shown in our book. Okay, then there is a region called D region, and what's the exact function of D region is not known. Rep1 and Rep2, they are used for replication, and obviously origin of replication, we know the point to start the replication. Okay, now keeping in mind of this map of 2 micrometer plasmid, let's just see which kind of vectors are developed for yeast, and this actually is our fourth learning objective for today's lecture, that which type of vectors can be used with yeast as a host. Okay, now there are three main classes of yeast vectors: the plasmid vectors, like shuttle vectors, and these are yeast episomal plasmids, yeast replicative plasmids, and yeast centromere plasmid. The two of these, yeast episomal plasmid and yeast replicative plasmids, are present in our chapter 7 from the book T.A. Brown, while this one, yeast centromere plasmid, I have taken from another source. Okay, then comes vectors that integrate into the yeast chromosome. So obviously that the name is yeast integrative plasmid, and this is also present in your book. Then yeast artificial chromosome, and yeast artificial chromosomes are the most important vectors developed on the base of yeast chromosome, on the design of yeast chromosome, and it is however not so convenient for use as expression vector. It is basically used to construct the genome libraries. Okay, so let's just discuss these vectors one by one. So first we are going to discuss this yeast episomal plasmid. Okay, now this yeast episomal plasmid, actually the word episome comes from, what do we mean by episome? Episomes are the genetic materials, or you can say the chromosomal material, which can exist independently of the genome, or plasmids, or they can get integrated into the genome. So they can replicate independently, as well as they can get integrated into the main genome and live as an episome. So yeast episomal plasmid, YEPs, these are shuttle vectors, and they are hybrid. That is, they are made up of some pieces of PBR322, some portion from yeast 2 micrometer plasmid, and a part of yeast chromosome. And also from the yeast chromosome, we are using a metabolic gene LEU2 as a selectable marker. So let's just have a look at this vector. So you see, half of it is composed of PBR322. We have studied the PBR322, and we know that it has an ampicillin resistant gene, a tetracycline resistant gene, and then you see this gray portion is the origin of replication taken from the 2 micrometer plasmid, and here again some portion from the 2 micrometer plasmid, and rest is having yeast chromosomal DNA portion having the gene LEU2. Okay, now LEU2 is a metabolic gene, and it is used as a selectable marker over here. Okay, now how we can use LEU2 as a selectable marker? So just first let us just see here. We have two selectable markers here for selection in E. coli. Okay, and then we have one selectable marker over here for selection in the yeast as a host. Okay, that's why it is called a shuttle vector. What is a shuttle vector? A vector that can be used in two different types of host is called a shuttle vector. Obviously, the shuttle vectors will have some portion origin of replication for both the hosts. Otherwise, it won't be able to replicate in both the hosts. Okay. Okay, now let's just see that how we are going to use LEU2 gene as a selectable marker in a yeast cloning experiment. So in order to use LEU2 as a selectable marker, first we need to ensure that we are selecting a yeast which is deficient in this gene, which is a LEU2 negative mutant. Such mutants are called auxotrophic mutants, the mutants which lack one or more metabolic gene, and they require a special kind of media to be grown. They need a media to complement for the gene which they are missing. For example, here we are having a LEU2 negative mutant yeast, and this LEU2 negative mutant yeast does not contain LEU2 gene in its chromosome. Okay, now so what we are going to do here, we are going to take media which contains leucine so that such kind of host can grow. So what is the requirement for growing a LEU2 negative yeast? Either we provided a media which contains the leucine, or we transform it with a vector that contains the LEU2 as a gene. Okay. Now here you see we are using a vector like we saw the yeast episomal plasmid. It has a portion of chromosome that codes for the gene LEU2. This means if we put this vector into this host, and the host which gets really transformed would be able to survive on a minimal media without leucine, and vice versa. So you see, if we put this vector inside this host, so this will not be LEU2 deficient anymore. It will have a LEU2 gene complemented by the vector it is having. Okay, so it can grow on a media without leucine. Okay, so this will form our basis of selection. Okay, let's just study the cloning experiment with the YEP13 vector. So the standard cloning procedure with YEP13 vector involves two steps. First, there is a primary screening and selection on an E. coli host, and then there is sub-cloning in a yeast host. Okay, let's just study step by step. So here, for example, when we did all our restriction, digestion, ligation steps, and we got, for example, we got these three vectors. One of these is self-ligated. The other one contains gene, it's the recombinant vector. It contains any gene. And the third one contains our gene of interest. Okay. Now, when we are going to do the transformation in E. coli, and we are going to grow the transformed E. coli on an ampicillin resistance medium, so you see that the E. coli which have got any of these plasmids would be able to grow on a media that contains ampicillin. Okay, now we will do the initial screening and selection, and we will come up with our desired clone, the clone which contains our gene of interest. Now we are going to isolate the plasmid from this clone, and we are going to perform a sub-cloning in yeast, in our yeast. Which yeast? The auxotrophic mutant yeast. Okay, now once we transform our yeast, then we are going to select the transformed yeast onto a minimal media that does not contain leucine, as we are using LEU2 as a selectable marker. Okay, so the cloning in with a YEP13 vector has two steps. First, the initial cloning in E. coli to select the recombinants, and then after we get our recombinants, we just do the cloning in yeast host. Okay, now let's just have some advantages and disadvantages of the yeast episomal plasmids. So here you see there are several advantages of the yeast episomal plasmid, as this plasmid lives as an episome, that is that it can live independently of the main chromosome, or it can get integrated into the chromosome. So if it is living independently, so we can see here that this can have a high copy number, which is 50 to 100, and this is fairly good. Okay, the second important thing is that it has high transformation frequency, means only little amount of the recombinant plasmid would be required to have more recombinant yeast hosts. Okay, now it is stable, fairly stable, and stable than the other yeast plasmids which we are going to study, and it's very useful for studying the complementation. Now, what is complementation? Just we saw in the previous slides that we are using an auxotrophic mutant yeast host. Now, this auxotrophic mutant yeast host does not have a LEU2, while our vector has a LEU2 gene. So, our vector is complementing the auxotrophic mutant host. Okay. Now, another advantage of the YEP is that it can be readily recovered from the yeast. Means if we want to get the vector out of yeast host, we can easily recover it. Now, beside this, there are two main disadvantages. I will first discuss this one and then I will come to this one. So novel recombinants are generated in vivo by recombination with endogenous 2 micrometer plasmid. Now what do we mean by this? We know that yeast as a host contains 2 micrometer plasmid, the natural one, not our vector, the natural one. So if we have auxotrophic mutant yeast as a host which already contains a 2 micrometer plasmid, and then it is transformed with our YEP vector, then what will happen? The YEP vector will have recombination with the 2 micrometer plasmid and some novel recombinants would be generated. Now, actually, this is something which we don't require. Okay. The second thing is homologous recombination. Now, what is the disadvantages of this homologous recombination? We're just going to see in the next slide. Okay. So, what actually happens here that suppose this is our vector. Indeed, it is a vector. If you see here, it has LEU2 gene. Okay. Now this is our YEP13 vector. We didn't put anything else. Just we are showing the LEU2 gene here so that we can understand what homologous recombination is and what is the consequence of this homologous recombination in this case. So this is our yeast chromosomal DNA. And this all is happening once the vector goes inside the auxotrophic mutant host. Obviously, the auxotrophic mutant host does not contain a fully functional LEU2 gene and it has a mutated LEU2. Okay, but it has some portions of LEU2 here and here which are actually matching our LEU2 gene in our vector. So, the LEU2 gene in our vector can have a homologous recombination and as a result, the whole vector can get integrated into the host chromosome. Okay. Now, the host chromosome will have two copies of the gene, one the mutated one and the other one which contains the whole vector and a fully functional gene. Now, what will happen now? So the whole vector is integrated into the host chromosome and this is trouble. Why? Because now if we want to get back our vector, our recombinant vector, we will not be able to get it back because it is already integrated into the host genome. Okay. Now, let's come to the second type of plasmids, and these are yeast integrative plasmids. Okay. Now, yeast integrative plasmid, the name indicates that they are actually purposely designed to get integrated into the yeast chromosome. Why do we want it to integrate into the yeast chromosome? Because we want, first thing, a stable transformation. A stable transformation means it will let our gene of interest to be integrated into the yeast and make the yeast as a transgenic. Okay, so yeast integrative plasmid, what they basically are and how they are made, they are basically bacterial plasmid that can insert themselves into the DNA of one of the yeast chromosomes. Okay, now the genes which are integrated into the yeast chromosome are less liable to be lost, mean they have a stable transformation and they will divide only when the cell is going to be divided. Okay, so by division I do not mean by dividing 1 into 2, it's mean like replication. Okay, now although transformation efficiency of yeast integrative plasmid is low and the copy number is 1, it has proved to be very useful in yeast genetics. Okay, yeast integrators are used for placing the DNA segments within yeast genome. Okay, so this means if we want to make a transgenic, we are going to use yeast integrated plasmid. Okay, now they can be replicated and maintained in E. coli, but not in yeast. Okay, they can be replicated and maintained in E. coli, not in yeast. What does this mean? This means that if you want to replicate and grow them as a vector, as an independent vector, they can be done. This can be done in E. coli, but once it goes to the yeast, it gets integrated into the genome, and then you will not be able to maintain it as a separate vector. Now it has a transformation frequency of 10 is to power 4 transformants per microgram of DNA. Okay, so obviously the transformation efficiency is not very high, but still it is very useful. So here is the typical map of a yeast integrative plasmid YIP5. You can see it has ampicillin resistant gene, tetracycline resistant gene. The size is 5.5 kb, which is fairly small, and it has a URA3 gene. URA3 again is a yeast metabolic gene, and this is involved. It is an important gene in the metabolism, and we are just going to see what. Okay, so we will just see in a bit that what is the function of this URA3 and how it is going to help us. Okay, first let's have a look at the advantages and disadvantages of YIP, the yeast integrated plasmid. The yeast integrated plasmid is having a stable transformation, so it gives the most stable maintenance of a clone gene inside a transgenic yeast. Okay, now, it also behaves as an ordinary genetic marker, and it is useful for surrogate genetics of yeast, like to introduce deletions and inversions. So we can use this to cause the deletions and inversions in the yeast. Okay, what are the disadvantages? As we studied in the previous slide, the disadvantage is that it has low transformation frequency. Okay, and also that if we want to recover this plasmid, so what we need to do, we need to cut the chromosomal DNA, and then we will have to retrieve our clone gene and our original vector. Okay, now, let's just have a look that how URA3 is working as a selectable marker. So once we are using a competent yeast cell, which yeast cell here we are using? We are using the auxotrophic mutant that lack the URA3 gene. The URA3 gene is involved in the metabolism of uracil. Okay, so we know that uracil is an important nucleotide. Okay, and it is used in transcription. So once we are incubating these plasmids with which contain the URA3 along with the competent cells, which are auxotrophic mutants, the deficient mutants which do not have URA3 gene, and then after transformation we are growing them onto a media, selective media, and this selective media does not contain uracil. So this means only those yeast would be able to survive which are transformed now, which have already taken plasmid containing the URA3. You can see this and you can read more about this on this website. Okay, this has been taken from the Sigma Aldrich protocols. Okay, then comes the third type of plasmid, which is present in yeast, and it is yeast replicative plasmid. The name is telling us something, that this plasmid, what is the purpose of this plasmid? To replicate, to make more copies of our gene of interest. Now, these plasmids were first constructed by Strohel in 1979, and it contains 1.4 kb yeast DNA fragment that contains another metabolic gene, TRP1. Now, TRP1 is a gene which codes for an enzyme which is involved in tryptophan biosynthesis. Okay. Now, it also contains portions of PBR322, and these plasmids, they are made in such a manner that they do not integrate into the yeast chromosome. Okay, now they can maintain, although they have low copy number, but they are very useful. Like they contain 1 to 20 copies per cell, and they carry an ARS, the autonomous replicating sequence, and this allows them to replicate when the cell divides. Okay, now their transformation efficiency is really high. High frequency of transformation, that is 10 to the power 4 microgram per DNA. Yeah, it is high as compared to other plasmid, but we will see another plasmid which has more transformation frequency than this one. Okay, now let's just see a typical yeast replicative plasmid. A typical yeast replicative plasmid, it contains tryptophan synthesis gene as a selectable marker. It contains portions from the PBR322. Again, it's a shuttle vector. It can be used in E. coli and yeast. It has ampicillin resistant gene, it has tetracycline resistant gene, it will have a region of replication for the E. coli, and it will have autonomous replicating sequence for replication inside the yeast host. Okay, now let's just see here. First, we are going to see how we are going to use this TRP as a selectable marker. Obviously, we are going to have a competent auxotrophic mutant yeast lacking the TRP1. We're going to incubate it with the plasmid which contains the TRP1 gene, and then we are going to select it on a media that lacks tryptophan. Okay, now you see only those yeast cells would be able to grow here which contain the plasmid or which are transformed. Okay, now let's just see some advantages of this plasmid. So they are easily recovered from yeast. Why they are easily recovered from yeast? Because they do not integrate into the yeast cell. Okay, now it has a high copy number, although we saw normally it maintains 1 to 20, but special arrangements can make or increase the copy number, which I'm going to discuss in the next lecture. Now it has high transformation frequency. The transformation frequency is comparable to that of the previous yeast episomal plasmid, but it is still higher than the other plasmids. Now very useful for complementation studies, same as the yeast episomal plasmids, and it can be integrated into the chromosome if desired by making again the use of TRP1 for homologous recombination. And the only one drawback here, the transformants which we get by using such vectors, they are not very stable. Okay, now let's move on to the third type of yeast vectors based on the yeast 2 micrometer plasmid, and again this is a shuttle vector and it contains, see, yeast centromere plasmid. It is little bit different than the previous plasmids which we studied. And what is the difference here? The difference is it contains a clone yeast centromere. Okay, now so what happens during the chromosomal replication? This yeast centromere is actually helping it to move. Okay, now chromosomal replication region is similar to yeast replicative plasmid, that is, it contains autonomous replicating sequence. It has a transformation frequency similar to the other plasmid. Okay, and the centromere plasmids are maintained at 1 copy per cell, just like small circular chromosomes. Okay, so they do not have more copies. So let us just see this yeast centromeric plasmid here. It has autonomous replicating sequence, it has centromere, and let me just use this pointer. It has centromere, origin of replication, ampicillin resistant gene, okay, and it has a yeast selectable marker over here. This yeast selectable marker can be any gene, any metabolic gene from yeast. Okay, now they have an advantage. What is the advantage? That these can be stably maintained. But they have several disadvantages. What are the disadvantages? Obvious disadvantage. It has a low copy number, and the viability is reduced. The cell viability, which cell? The cell which harbors this plasmid. And why the viability is low? Because the yeast cells are forced to maintain multiple yeast centromere plasmids, okay, and bearing independent selectable markers. So for this reason, the overall viability of the yeast host containing yeast centromere plasmids is low. Okay. So till now we have studied the plasmids based on the 2 micrometer plasmid and portions of the yeast chromosomes and PBR322, all shuttle vectors. Now we are going to study another type of vector that can be used with yeast, and that is an artificial chromosome. Now the name artificial chromosome tells us that it's not like a plasmid, it's a normal linear yeast and yeast chromosome. Okay, so there are certain features of this, like you see, like if we have a look at this diagram, so we see the normal structure of a chromosome. The chromosome contains 2 telomere at the ends, okay, like these ones, and it has a centromere in the center and several origin of replications. Why do the chromosomes have several origin of replication? Because chromosomes are big. So to start and complete the replication within fractions of seconds, so we need several origins of replication. Similarly, a yeast artificial chromosome is having a structure similar to a chromosome. Okay, it has 2 telomeres which are actually preventing the artificial chromosome from being degraded by the exonucleases, and these are also involved in the chromosomal replication. Okay, so they have origin of replication as that of a plasmid, which we are going to see in our next slide. They have centromeres which help in proper segregation of the chromosome. They have selectable markers, and they are usually maintained as single copy per cell, and they are actually the hybrids from the plasmid DNA and the yeast DNA. And they grow where? They grow in yeast. Now let's just have a look at the yeast artificial chromosome. So this is one of the earliest yeast artificial chromosome. It has been taken from your book Gene Cloning and DNA Analysis by T.A. Brown, page 111. Let's just here. So let's start with the origin of replication. So it has origin of replication here. It has been taken from the yeast 2 micrometer plasmid. Now besides that, it has a centromere region. Okay. Now it has TRP1, which is a selectable marker coding for enzyme involved in tryptophan synthesis, URA3 involved in uracil synthesis. It has 2 telomeres, okay, and these are flanked by a sequence which has 2 restriction enzyme sites of BAMH1. Okay, so this means if we cut this plasmid with BAMH1, it will become a linear chromosome, which we are going to see in a minute. Okay, now here we have another gene which we can see only here in the yeast artificial chromosome and not other yeast vectors which we studied so far. So SUP4 gene. What is the importance of this SUP4 gene? This SUP4 gene codes for a red pigment formation. This means any transformed yeast cell which is transformed by this vector will be able to produce red pigment. So the colony will look like a red colony. Okay. unless until we have a recombinant plasmid. So you see there is a restriction enzyme site SNAB1 in the middle of this gene SUP4. This means that we are going to use this gene for cloning purposes, and we are going to insert our gene of interest by cutting with SNAB1. Okay, now here you have two restriction sites, BAMH1 and SNAB1. These BAMH1s are not used for cloning, they are just used to linearize your yeast artificial chromosome, while the SNAB1 is used to clone your gene of interest, okay, which we are going to see in a minute. So, the advantages of yeast artificial chromosome, there are several advantages. And what is the biggest advantage? It can have a large insert. Okay, so these vectors are highly stable and they mimic a chromosome, so they will behave like a chromosome, and larger genes can be cloned in it. So it has helped us to study larger genes like that of gene for muscular dystrophy. Okay, now disadvantage is the inserts are very unstable. Okay, and why unstable? Because of intramolecular rearrangements. Okay, intramolecular rearrangements do occur within the chromosomes, and this leads to insert instability. Now, resolution is difficult once introduced into cell. Once you introduce this yeast artificial chromosome into the cell, it is difficult to recover. You know, looking at the big size of this, yeah, it is difficult to recover. Big size, not the big size of self-ligated vector, the size of recombinant vector. The recombinant vector becomes fairly big because it can clone like 600 kb to 1400 kb fragment. Okay, and obviously it cannot be mapped by standard techniques which are used. Okay, now let's just see how do we clone in a YAG vector. So let us just concentrate on the restriction enzyme sites. Here is the restriction enzyme site for BAMH1, here is another one, and here we have for SNAB1. Okay, so we know that we are going to clone here, and these two we are just using to linearize our vector. So what do we do first? We restrict them with BAMH1 and SNAB1. So BAMH1 will remove this flanking, they're flanking this stuffer fragment. It is like, stuffer fragment which we studied in case of lambda replacement vectors. So similar to that, it is a stuffer fragment. Okay, so once you cut it with BAMH1, this portion will be removed and the whole yeast artificial chromosome will become linear. Okay, now after that, when we cut with SNAB1, so it is actually present inside this gene SUP4 that goes for a red pigment. So you see the gene will become truncated and non-functional. This will lead to now once you clone your gene of interest over here, so you see the insert DNA will be taking the place in the middle of your SUP4 gene. As a result, the SUP4 gene will become insertionally inactive. Okay, now here you see this whole structure represents a chromosome. Okay, it's like a chromosome. It has centromere, has region of replication, it has telomeres at the end, and it has essential elements of a vector, the selectable marker, the restriction enzyme site, and a big gene inserted. Okay, so obviously, as I just told in the previous slide, it can have a very large fragment. Okay, 600 kb to 1400 kb, and this is very important thing. Okay, it can reduce the size of human genome library to only 6500 clones. Previously, you see back vectors, the bacterial artificial chromosomes were used, and we need 30,000 clones from using back vectors to make human genome library, while the number of clones is fairly reduced to 6,500 if we are using a YAG vector. Okay, the second thing is most of the genes involved in diseases, they are big genes, and if we want to clone a whole gene, then obviously YAC is a vector of choice, and it has opened the way for studying functions and mode of expression of several genes, most of them the genes involved in diseases. Okay. Now, YACs can be used with the mammalian host. This is one of the very huge advantage of YACs, that we can clone big mammalian genes inside the YAC, and yet we can use YAC to transform the mammalian cell lines, and we can just have our gene in their native environment. So we will see their expression in a very native environment. Okay. These are used in large-scale DNA sequencing projects. Obviously, if our aim of gene cloning is to study a genome, obviously YAC would be really helpful. Okay. Now, transformation of yeast. Now, what do we mean by transformation? All of us, we know transformation is introducing a foreign DNA into a normal cell. Okay. Now, there are several methods which we have studied for E. coli, like you see, electroporation and calcium chloride method. So similarly, for yeast, there are several methods which have been reported: the lithium acetate treatment, the spheroplast technique, the electroporation, biolistics, and glass bead methods. Some of these methods, like biolistics and electroporation and spheroplast, they are present in your books, T.A. Brown, and the other one, the Nicole. And this glass bead method and lithium acetate treatment, I will teach you at the end of this chapter. I will teach you the methods for transformation of eukaryotic hosts. So there I will teach you in detail. So these methods are commonly used for our common yeast host, the Saccharomyces cerevisiae, and these are also useful for cloning in Schizosaccharomyces formidae, Candida, and Pichia pistorius. Okay, and it can be used with other filamentous fungi like Aspergillus. Here, here I have added a link from from a journal, and this journal is publishing the video papers. So you can just follow this link and you can watch a video on how transformation in yeast is carried out. It's really useful. Okay, here I have listed comparison of transformation method. I will not go into the detail. You know electroporation already. We are using electricity to create pores in the cell membrane, and then biolistics, our gene gun. So we are using the tungsten coated DNA particle, tungsten particles coated with our DNA, and then we are just bombarding into the nucleus of the cell. Agitation with glass beads, we are going to study it in detail at the end of this chapter, and vacuum infiltration and shock waves. All of these methods, here there is a comparison, and at the end of this chapter I'm going to show you videos regarding these. Okay. Now, so we have studied today that why we are choosing eukaryotic hosts, and why we are not using the E. coli, which is fairly simple to use and it can grow quickly. It can have a very high copy number of our clone gene. So why we shifted towards the eukaryotic host? Because we wanted to give a native environment to many of our eukaryotic genes, specifically when our aim of cloning is to express a gene or to make a protein. Okay. Now, so we studied today yeast as a host, and we studied the vectors which we can use with yeast as a host. Obviously, yeast is very simple organism. It is a eukaryote. It has minimal nutritional requirements. Its biology, genetics, and physiology is known. It's very good. It can be used with the bioreactors, or it can be used on the lab scale, but it also has certain limitations. What are the limitations? The generally low expression level. Now, expression level of gene is generally low in yeast. Okay. Then plasmids frequently lost during scale-up. While we are carrying out the scale-up processes, the procedures, the yeast cells tend to lose our plasmids, which we have seen in many of our yeast-based vectors, that they do not have a stable transformation and they are lost, okay, during the scale-up procedures. And another important thing is some of the secreted proteins. If we are using yeast cells to as a factories to produce our proteins, then some of the proteins, they get trapped in periplasmic space, and it makes the downstream processing very difficult. Okay, so these are some of the limitations, and that limitations actually give rise to the use of other hosts for eukaryotic genes, which we are going to see in our next lecture. Okay, so with this, I end up this lecture over here. Here are some of the references, and these are the sources from which I have taken materials to build up this lecture. You can review these, and you can revert to your book, Gene Cloning and DNA Analysis by T.A. Brown, and you have most of the details which I have discussed over here in this book. Okay, thank you very much.